The basic structure of a neuron consists of dendrites, the cell body, the nucleus and axon, and the synapse. A signal goes through the dendrites, down the axon, and to the synapse. In a neuron, the inside of the cell has a higher concentration of positively charged potassium and negatively charged proteins that give it an overall negative charge. The outside of the cell has a higher concentration of positively charged sodium that has an overall positive charge. The attraction between the opposite charges creates the membrane potential or the charge difference between the outside and inside of the cell. When a signal is not being sent, the membrane potential is between -60 and -80 millivolts because the inside of the cell is more negative. The positive-negative gradient is maintained by the sodium-potassium pump shown in green because it pumps 3 sodium out of the cell and 2 potassium into the cell, enabling the outside to be more positive than the inside.
When a stimulus is detected, it causes the ion-gated sodium channels to open, allowing positively charged ions to enter, which increases the membrane potential. If the membrane potential reaches -55 millivolts, it crosses a threshold that opens adjacent voltage-gated sodium channels that causes the membrane potential to reach positive 35 millivolts. This causes the action potential to occur and temporarily reverse the membrane potential as the signal moves through the axon.
When the membrane potential reaches 35 millivolts, it causes the sodium channels to close and the potassium channels to open. This positively charged potassium rushes out of the channels, which makes the inside of the cell negative back to the resting potential.
The membrane potential continues to decrease past the resting potential to around -80 millivolts. During this period, the sodium channels are deactivated, which means that if there is a second stimulus, the neuron cannot achieve another action potential. This prevents signals from traveling in both directions. The gated potassium channels close, and the sodium-potassium pump restores the concentration of the ions back to the resting potential.
In myelinated axons, the sodium channels are restricted to gaps in the myelin sheath called nodes of Ranvier. Action potentials occur more quickly in myelinated axons because instead of triggering one channel at a time, a current can leap from one gap in the myelin to the next.
In order to transmit information to other neurons, the presynaptic neuron releases a chemical neurotransmitter through the presynaptic terminal. The terminal holds thousands of synaptic vesicles containing the neurotransmitter. When the action potential reaches the terminal, it activates the voltage-gated calcium channels. The influx from the positive ions causes the vesicles to fuse with the cell membrane, releasing the neurotransmitters in the synaptic cleft. The neurotransmitters bind to postsynaptic receptors, which are ligand-gated ion channels that open to accept calcium and potassium. In an excitatory response, the neurotransmitters depolarize the membrane to push the neuron closer to the threshold.